We will now introduce our next speaker. So Máté, if you'd like to share your screen. Perfect. That's great. So to introduce Dr. Máté Manczinger, Máté graduated with a medical degree from the University of, I'm going to get this wrong, the pronunciation, Szeged?
Szeged.
Szeged. I'm
Yes.
learning in my Hungarian, University of Szeged in 2010,
Yeah.
and he pursued further studies in dermatology and immunology. By 2016, he completed his PhD and dermatology specialization, and he's become principal investigator at the Biological Research Center in Szeged, Hungary in 2022. Under his direction, the concept of HLA promiscuity has been thoroughly examined, resulting in significant publications in PLOS Biology and Nature Cancer. His latest work, in collaboration with Thomas Vogel and Sasha Zanakova, investigates the relationship between HLA promiscuity and antibody diversity. So, Máté, thank you very much for joining us today. And the title of your talk is Negative Trade-off between Neoantigen Repertoire Breadth and the Specificity of HLA Class 1 Molecules Shapes Anti-Tumor Immunity. So over to you. Thank you.
Thank you very much. So it's a great honor to talk about our results in this very nice symposium. So only some very basics. Of course, we know that the immune system has to fight against the harmful agents and has to tolerate the non-harmful ones. And during still the 1990s, it was believed that immune system has nothing to do with cancer. And Jim Allison said that the people laughed at him in conferences that he talked that the immune system indeed has something to do with cancer. So the immune system now, it's crystal clear that has an important role in defending our body against cancer. What is the basis of anti-tumor immune response? The basis of anti-tumor immune response is that mutations are constantly generated in tumor cells, and many of these mutations modify the sequence of proteins and the sequence of peptides that are generated inside the cells. And as all we know, the HLA molecules, especially HLA class 1, has an important role of presenting these mutant peptides on the surface of tumor cells. And if the T cells in the repertoire recognize these presented peptides and can induce a destructive immune response, then the immune system can defend us against cancer. Of course, the tumor cells has developed a strategy. Of course, it's not an active development, it's an evolutionary process. So these tumor cells overexpress negative regulatory molecules. For example, the PD-L1 that inhibits the activation of the T cell. And the cancer immunotherapy exploits this mechanism. The cancer immunotherapy inhibits this inhibition, so it releases the brake on immune destruction of the tumor cells. And it was highly effective also in late stage cancers. On this screen, you can see the previous standard of care treatment of non-small cell lung cancer and the survival probability in that case. And as you can see, the cancer immunotherapy, the pembrolizumab, improved significantly the survival of patients. So it was not a surprise that the two researchers who had a pivotal role in developing these therapies were awarded Nobel Prize in 2018. However, there are still patients, depending on the tumor type, the fraction can range between 20% to 70%, or even more, who don't respond to the cancer immunotherapy treatment. So it is essential to identify biomarkers. And there is a governing view in the field that it's, I would say, a quantitative view, which suggests that the higher number of mutations can be found in a given tumor sample, it is more likely that peptides will be presented on the surface of tumor cells. So if we release the brake on the immune system, it will be more likely that the tumor will be destroyed by the immune system. So we would like to somehow be more provocative and say that this quantitative view is not that perfect, and we would say that the quality of antigen presentation also matters. So in our research, we focuses on HLA molecules. These HLA molecules has extreme diversity, and consequently, it is quite unlikely that two random individuals will carry exactly the same HLA variants. And these different HLA variants are likely to bind different peptide sequences. And the different variants, as we know, are associated with susceptibility or protection from different diseases like infections, autoimmune disorders, and allergy. And there is an interesting feature of HLA molecules. This is the so-called peptide binding promiscuity. And it means that what is the number, what is the fraction of peptide sequences that can be bound by a given HLA allele? So what is the promiscuity of peptide binding by these alleles? And there are alleles that can bind only a small fraction of the peptide sequence space, and there are alleles that can bind a much higher fraction. And we call the former ones specialists and the latter ones generalist HLA molecules. And in one of our previous work, we suggested or we hypothesized that the generalist HLA molecules may bind a higher number of antigens or peptides of a higher number of different pathogen species. And we showed that in indigenous populations, the generalist alleles are more common in those geographic regions where the pathogen diversity is much higher. And now we are carrying out experimental validation. We are collaborating with a research group, and we aim to show that indeed in those individuals who carry generalist HLA alleles, we can find a higher number or a higher fraction of antibodies against people or against pathogens. And we are having quite promising results. But the focus of this lecture or this presentation is on cancer. So we thought that carrying generalist HLA alleles may also be beneficial in terms of anti-tumor immunity. So, and the rationale is simply the quantitative view. So if in a given tumor, that we can find a generalist HLA allele, it is more likely that the neopeptides that are generated inside the tumor will be presented on the surface of the tumor cell. To test our hypothesis, we examined 3 cancer immunotherapy cohorts, 2 melanoma cohorts and 1 non-small lung cancer cohort treated with different cancer immunotherapy agents. And we aim to determine the promiscuity level of HLA molecules carried by each individual. And we developed the method. I don't want to go into technical details, but this method was based on the peptide binding motif of the HLA molecules. And based on the motif, we were able to calculate the peptide binding promiscuity of these molecules. And we wanted to validate this method, and we used the proimmune, or we asked to use the proimmune reveal assays. We selected around 30 diverse, well-known neopeptides or neoantigens, and we asked to determine the binding of these peptides by 13 different HLA class 1 molecules. And we found that the calculated allele promiscuity level strongly correlated with the fraction of the bound neoepitopes by these HLA alleles. And then we wanted to see whether those people who carry generalist HLA alleles indeed survive better than those ones who don't carry this generalist HLA allele. But we found absolutely the opposite. We found that those individuals who carry highly promiscuous HLA alleles survived much worse than those ones who didn't carry these promiscuous alleles. And we were in trouble because we wanted to explain this finding, and we had quite a lot of hypotheses. One of the hypotheses came from enzymology. It was suggested that in case of promiscuous interactions, for example, in case of an enzyme and a substrate, if an enzyme is promiscuous, so it can bind a high number of substrates, then the complex between the enzyme and the substrate is less stable. And we thought that it might be also the case in case of generalist HLA molecules. So the generalist HLA molecules could bind less stably the epitope or the peptide. So the peptide MHC complex could be less stable. And we know that the stability of the peptide MHC complex is the prerequisite of the T cell receptor activation. And in order to test this hypothesis, we asked the ProImmune to carry out complete rate assays on those HLA molecule and now peptide pairs, which were found to be bound in the previous analysis with ProImmune revealed assays. And on these plots, you can see two groups of HLA molecules. The blue group shows the complexes between low promiscuity HLA molecules and their peptides, and the red group shows the values for complexes between highly promiscuous, so generalist HLA molecules, and neopeptides. And on the first plot, you can see the half-life of complex assembly, so how fast a complex is assembled in vitro. And as you can see, in case of promiscuous HLA molecules, the time needed for assembly is significantly lower. And on the right side, so in contrast, what we would expect, we can see that also the half-life of the complexes is higher in case of promiscuous HLA molecules. So it seems that in contrast with expectations, the promiscuous HLA molecules, the generalist HLA molecules are likely to form stable complexes with neopeptides. So we went further and we tried to find out what could be the explanation of our findings. And I want to show you the most likely one. So in order to understand this explanation, we first have to see what is needed for effective recognition of neopeptides by T cells, by the immune system. So in tumor cells, if we have a mutated peptide, this mutated peptide very frequently differs from the original one in only one amino acid. And for every neopeptide, we have the normal original non-mutated neopeptide, probably generated in healthy cells. And it is ideal if the immune system only meet with the mutated peptide, so the neopeptide, during life. And if it's not ideal, if the immune system meets both with the healthy cells or both with the normal peptide and the mutated peptide, because the T cells are cross-reactive. And if there is only a very low difference between the normal peptide and the mutated peptide, this is quite likely that the T cell will respond with tolerance against the original peptide, and that it will respond also with tolerance to the mutated one. And so the optimal case is when only the mutated peptide is presented on the surface of the tumor cell, because it's likely that the T cell will respond with destruction. And we thought that in case of generalist alleles, as these alleles are non-selective, they are generalists, they also likely to present the non-mutated original peptide and the null peptide, while in the case of specialist alleles, only the mutated peptide will be presented. There are measures developed in the literature to explain this differentiation capacity based on the binding of the original and the mutated peptides by a given HLA allele. And we tested what is the capacity of given HLA allele to distinguish between the mutated and the original, or not the allele, of course, but the immune system, to distinguish between the original and the mutated peptide. And we found a strong negative correlation between the promiscuity of HLA molecules and this distinction capacity. And of course, this is only a bioinformatic approach, a computational approach yet. So we wanted to also see that in those tumor samples which carry the generalist HLA molecules, there are signs of immune tolerance. And it was exactly what we saw. So in the microenvironment of these tumors, we saw the signs of intensive immune tolerance, high level of cancer-associated fibroblasts, and so on and so on. In sum, we can say that, of course, these generalist HLA molecules has a beneficial role. That's why they are prevalent in human populations. They are better in antiviral immunity. So they confer protection from a higher number of viral species. But as a negative trade-off, they come with worse anti-tumor immunity. Thank you for your attention, and I would like to thank my group who helped in this project. Thank you.
Thank you very much, Máté. That's very clearly explained, and it's this really fascinating hypothesis and the data that you've generated from it. Can you just go back up one slide? If anyone has any questions, please feel free to raise your hand. Yeah, thank you. So I guess a question here is that linking this with one of your earlier slides where you were talking about the generalist HLA alleles in maybe more the populations, the sort of tropical zones where you've got a lot of pathogen exposure. Yeah, thank you. Then does that then suggest that there's a higher level of tumor or lack of tumor control in those populations, or actually is that very difficult to determine because of the, I guess, the more Western lifestyle differences that there are in the more specialist allele areas, as it were?
Yes, absolutely. So it's a question that we would like to validate somehow, but still we have very hard times with it. So if in these populations in the tropical regions, or those individuals who carry generalist HLA molecules, is the chance of having a tumor at all is increased, or we can only see this negative effect of carrying generalist molecules in case of cancer immunotherapy. So we are working on it. We are checking these things in large biobanks. But what we see that we have to control for such a high number of factors that it's very hard to show a clear trend. There are small, so not that strong trends, but I would not say yet that yes, indeed, carrying a generalist HLA molecule is associated with increased cancer risk.
Yeah, I can appreciate that's a very difficult thing to demonstrate, but good luck. That'd be good.
Thank you.
Also, in regards to the different allele, or your definition of generalist versus specialist alleles, are the definitions of that in some of the publications that you've referenced there, or how are you? Because it's something that we obviously see when we're doing these binding assays. We very, all the time, if we're screening a set of peptides, we'll always see that some alleles, by nature of the binding motifs, will have multiple hits, and then others have very, very few, and just the odd one. So it's very much in line with what we observe in the physical assays. But it's quite interesting to sort of be able to put a label on that, and that's something which I've not really come across before. Seeing your work, I quite like it because it kind of explains what we actually observe. So it's nice to have a label on it, but I'm just wondering how you're defining it.
Yeah, so in case of generalist and specialist alleles are just some arbitrary classification. So I would say that I use this classification to make the presentation much clearer. But I would say that this is a continuous scale, and of course, on this continuous scale, we can create arbitrary classes. Here on this plot, we also made some arbitrary classification based on quantize, but we have to see that this is a continuous scale. So this HLA promiscuity is a continuous scale, and we cannot say that this molecule is a generalist and all of others are not.
Yes,
It's
that makes
just
sense.
for the explanation.
Yeah, well, it helped because it was very clear. So thank you. Great. So I think we have to move on to our next speaker now. But yeah, thank you very much indeed, Máté, for your presentation. That's great.